Aquaculture is one of the fastest growing food production sectors, with tilapia being widely cultured due to its rapid growth and adaptability. Feeding management plays a crucial role in maximizing group performance and feed efficiency. Feeding frequency in particular influences nutrient absorption, metabolism, and overall fish health. However, inappropriate feeding schedules may lead to poor growth or fish wastage. Therefore, the study aims to evaluate the effect of different feeding frequencies on the growth or performance of tilapia under a controlled feeding schedule.
Here is the problem statement of our research, which is feeding imbalance. Basically, in aquaculture, feeding practices are really important to make sure fish grow well and production stay high. But one common problem that farmers face is not knowing the right feeding frequency, which can cause either overfeeding or underfeeding. When fish are overfed, it increases feed costs and also affect water quality because of uneaten feed. On the other hand, underfeeding can slow down fish growth and reduce productivity. Even though we know feeding frequency affects growth performance, there is still some uncertainty about what feeding frequency works best under controlled conditions. So, this study is needed to evaluate different feeding frequencies and find more efficient and sustainable feeding strategy for tilapia culture.
Move on to research objectives. Firstly, to evaluate how different feeding frequencies affect the growth performance of tilapia in aquaculture by measuring weight gain and length. Secondly, to assess the survival rate of tilapia under different feeding schedules to ensure sustainable aquaculture production.
For the H-null, which is null hypothesis, there is no significant difference in the growth performance of tilapia under different feeding frequencies. And for the H1, which is alternative hypothesis, different feeding frequencies significantly affect the growth performance of tilapia.
Research question. How do different feeding frequencies affect the growth performance and survival rate of tilapia under controlled aquaculture conditions in achieving efficient production?
Significance of this research. Good day everyone. For my part, I will explain the significance of this research. This study is important because it helps improve efficiency in tilapia farming. Feed is the highest cost in aquaculture, so feeding the best feeding frequency can help reduce the feed wastage and save costs. Besides that, proper feeding can improve fish growth, survival rate, and overall health. It also helps maintain the better water quality. Lastly, this research is useful for students, researchers, and farmers. It can use as a reference and help us support more sustainable aquaculture practices.
Now, we are going to talk about the literature reviews. After doing our research, these are the topics related to our studies.
So, let me start with what these studies actually did. AGR240 back in 2010 worked with African catfish, testing how often they should be fed, from once up to 4 times a day, and look at the growth, feed efficiency, and profit. Then, Pota in 2016 studied juvenile tilapia in low salinity water, feeding them between 2 and 5 times daily, and measured growth, feed use, and body composition. Kaya and Bill Govan in 2005 compared 1 to 3 to 6 meals per day in young tilapia, focusing on growth and feed conversion. Meanwhile, Bila in 2010 took a different angle, looking at tilapia and carp in rice fish farming, testing feeding schedules from 1 to 5 per week, and even measured rice yield. Finally, Firdaus in 2014 studied monosex tilapia fry, fed 3 to 4 or 5 times daily, and checked growth, feed conversion, and survival. Now, what did they find? AGR240 showed catfish grew best at 3 meals a day, with no extra benefit at 4. Pota found tilapia juvenile had the highest weight and feed conversion when fed 4 to 5 times daily. Kaya and Bill Govan saw growth improve with more meals, with the best live weight at 6 meals, but surprisingly, the lower feed conversion ratio at just 2 meals. Bila's rice fish study showed tilapia and carp grew best with 5 feedings per week, twice daily, and this gave the highest net return. Firdaus found tilapia fry feed 5 times daily had the best growth, lower feed conversion, and highest survival. When we compared them, there are clear similarities and differences. All agreed that feeding frequency strongly influenced growth and feed efficiency, and survival was generally high. But the optimal frequency varied. Catfish did the best at 3 meals, tilapia juveniles at 4 to 5, fry at 5, and rice fish system at 5 meals per week. Some studies like AGR240 found diminishing returns at higher frequencies, while others like Tilapia and Firdaus saw continued improvement with more meals, so the effects really depend on species and age. Well, these studies had strong points. They were controlled experiments with replicates, which makes them results reliable. They measured multiple growth indicators like weight gain, specific growth rate, feed conversion, and protein efficiency. Aderolu and Bila even included economic analysis, which is very practical. Pota and Kaya look at body composition, giving insight into nutrition. Firdaus reported survival data, which adds credibility. Altogether, these strengths make the findings useful for aquaculture practice.
But of course, there were limitations. One trial was short, lasting only 28 to 78 days, so they don't know long-term outcomes. They were done in specific environments like low salinity water, rice fish system, or lab tanks, which limit generalization. Most focus only on early life stages, so we don't know much about market size tilapia. And not all studies included economic analysis, which is important for farmers. These limitations show where more comprehensive research is needed. There were also inconsistencies. The optimal feeding frequency differed depending on species, age, and system. Kaya found the best feed conversion at lower frequency, while Firdaus and Pota found higher frequency more efficient. Catfish growth plateaued at 3 to 4 meals in Adero study, but tilapia fry kept improving with more meals in Firdaus work. These differences suggest feeding strategies must be tailored to species and productive system. Looking at the gaps, we see that long-term trials are missing. Most studies don't follow tilapia from fry all the way to the harvest size. Few combine economic analysis for biological performance, except Adero and Bila. Research on environmental impact like water quality and waste is also limited. And because many studies focus on specific conditions, we don't know how broadly the findings apply. Filling this gap is key for sustainable aquaculture. And this is where my study comes in. By testing controlled feeding strategies in tilapia, I can clarify the optimal frequency under standardized conditions. My work is based on evidence that optimal frequency differs by age and system, addresses conflicting findings on whether higher frequency always improve growth, and links biological performance with economic outcomes. In short, my study fills the gap in consistency and economic analysis and provides practical recommendations for tilapia farmers.
Research design. In order to address the research question in this study, an experimental method based on completely randomized design CRD was used. All experimental units were maintained under the same environmental conditions to ensure consistency. The treatments were randomly assigned to each tank so that every unit had an equal chance of receiving any treatments. The use of CRD is suitable because the experimental units are homogeneous and allow fair comparison between the treatments.
The study was conducted in a controlled aquaculture tank system to ensure stable environment conditions. Tilapia fingerlings of similar size were used as a test species due to their fast growth and adaptability. The fish were randomly distributed into tanks to ensure consistency and reliability of the result.
Materials. For materials, we are using tilapia fingerlings which are healthy and uniform in size. Then, commercial pelleted fish feed which consist of fish meal, breed products, fish oil, vitamins, and binding agents. Next, we need aquaculture tank and aeration system like air pumps and air stones. Next, hand net for handling tilapia safely. Digital weighing balance for accurate result. And then measuring board to measure the fish length, water quality testing kit including pH meter, thermometer, and dissolved oxygen DO meter. Treatments. with 3 feeding frequency treatments T1, T2, T3. All tanks were kept under the same conditions such as water quality, temperature, stochasticity, and type of feed. The only difference between treatment was how often the fish were fed.
The experimental procedure. First, tilapia fingering will be stocked into the tanks or ponds at the same density for all treatment. This is important to ensure comparison. Then, the fish will be divided to 3 groups based on feeding frequency. Treatment 1 will be fed 4 times per day. Treatment 2 will be fed 3 times per day. Treatment 3 will be fed 2 times per day. All fish be fed using same type of commercial fed and based on the percentage of their body weight. The experiment will be conducted for several weeks, and throughout the study, the fish will be monitored regularly to ensure they remain healthy and active.
Moving on to data collection, data will be collected on a weekly basis throughout the experimental period. Growth performance will be measured by recording the initial and final body weight of the fish using a digital weighing balance. Weight gain can be calculated to compare growth among different feeding frequency treatments. In addition, survival rate will be determined by counting the number of fish remaining at the end of the experiment. Water quality parameters such as temperature and pH will also be monitored to ensure suitable condition for fish growth. Each treatment will be conducted in triplicate to provide replication and improve the reliability of the data. Replication allows for more accurate comparisons and supports statistical analysis to determine significant differences between treatments.
The data analysis. All collected data such as weight gain, survival rate, feed conversion ratio will be analyzed using analysis of variance or ANOVA. This test is used to determine whether there are significant difference between the treatments. If the results show a significant difference, which means the p-value is less than 0.05, is post-hoc tests such as the Taki test will be used to compare between groups. This analysis help us identify which feeding frequency gives the best growth performance.
Moving on to the last part, which is the ethical and practical. All procedures will follow proper animal welfare practices. Fish will be handled carefully to reduce stress. Water quality and aeration will be maintained throughout the experiment. In addition, this study is designed to be simple and cost-effective, making it suitable for small-scale aquaculture.
Oh!